Namaste friends, welcome back to Biology Center for True Learning. In this video, we talk of another important aspect from the genetics chapter, that is linkage. Linkage. So we begin the class with the meaning of linkage. What is meant by linkage? Some genes tend to inherit together. Some genes present on the homologous chromosome, so always like two, so enters the same gamete and also enters the same zygote. That is called a linkage. As the name indicate, two genes present on the homologous chromosomes are located closer to each other. So let us talk about the T.H. Morgan, who did the experimental, who proposed the experimental genetics. So it is necessary to have basic knowledge of linkage. For the first time, two scientists, viz. W. Bateson and Punnett, seen the linkage in the plant name Lathyrus odoratus. W. Bateson and Punnett observed or seen the linkage phenomenon in the plant Lathyrus odoratus. So it is the sweet pea plant. Here I make you remember the law of independent assortment given by the Mendel. So according to that principle, the genes present on the chromosomes so independently separate and assorted into the gametes. This is what we learned from that principle, right? That principle becomes right when two genes present on two different chromosomes. Then the independent assortment will be easy. If the same two genes present on the same chromosome, not on the different chromosome, the independent assortment may be or may not be possible. Regarding this, these two persons talked about. So let us see. They performed two experiments on the Lathyrus odoratus. Based on their experiments, they formulated hypothesis. Formulated the hypothesis of one is copulation, another one is repulsion. Copulation and repulsion are two aspects given by Bateson and Punnett from their experiments. So let us understand the experiment first. Before that, I would like to explain what is copulation and what is repulsion. The copulation is nothing but the genes come from same parent, enters the same gamete. That is called coupling. For example, so both dominant genes or both recessive genes present on the chromosome, so enters the same gamete, and enters the same zygote also, right? This kind of arrangement is also called a cis, right? The copulation is also called a cis. Next, repulsion. The dominant genes or recessive genes come from different parents, tend to separate and enters the different gametes. So let us understand with the picture. So this is the chromosome contain one dominant allele, one recessive allele. The another pair of homologous chromosome contains one recessive allele, another one is the dominant allele. So this kind of arrangement is called trans. So copulation is also called cis, repulsion is also called trans. Copulation means the genes come from the same parent, enters the same gamete and also enters the zygote. That is called copulation. So copulation in general, copulation means the closing together of the gene. Come closer together. The repulsion means get away from each other. so go away from each other. That is called a repulsion. So in case of copulation, we can observe the strong closeness between the genes. So in case of repulsion, so we can observe the separation of the genes and enters the different gametes. So these two concepts were proved by their experiments. So let us see. For their experiments, they selected Lathyrus odoratus sweet pea plant. Totally they did 2 crosses. For their experiment, they selected 2 characters in that plant. The one is flower color, another one is the pollen shape. Characters selected by them, one is flower color, the another character is shape of pollen grain. In the flower color, so one is blue color, so another one is the red. Blue color is the dominant over the red, that is recessive. Then shape of pollen, long pollens, then round pollens. Long is the dominant trait over the recessive trait, that is round. So these are 2 characters identified and selected by the persons for their experiment. So let us see the crosses. So in first cross, they selected 2 parents. In first cross, they selected 2 parents. This refers to the parent generation. So one parent with the blue color, long pollen producing plant. Another plant is red color, round pollen grains producing plant. The genotype of these plants are like this: capital B, then capital L, capital L. This plant genotype becomes small b, small l, small l. The gametes produced by these plants containing capital B, capital L alleles. The gametes produced by this plant containing small b, small l alleles. So when cross is made, cross pollination is done, they receive the F1 generation hybrids. They got capital B, small b, capital L, small l. This is the heterozygous blue color long pollen grains producing plant hybrid. Then they go for test cross. Already we explained what is test cross. Test cross means the cross between say F1 hybrid and the homozygous recessive parent. That is called a test cross. The test cross technique was actually proposed by the Mendel. So what are the test cross results Mendel obtained in the monohybrid and dihybrid cross? So here I wrote only dihybrid test cross by Mendel, that is 1:1:1:1. So this is the test cross ratio in the dihybrid experiment. So let us see whether these 2 persons, Bateson and Punnett, received the same test cross results or different test cross results. So this experiment will answer our question. Now we do the test cross between F1 hybrid and homozygous recessive parent. We know how to write the gametes. This parent produces 4 different types of gametes, like capital B, capital L, one gamete, one type of gamete, then capital B, small l, another type of gamete, then small b, capital L, third type of gamete, then small b, small l, another type of gametes. But this plant produces all the gametes with the small b and small l allele. Then, so when when the cross is made between, so these gametes, the results are like this. The test cross ratio here is 7 is to 1 is to 1 is to 7. This is the test cross ratio obtained by the Bateson Punnett in their experiments. So this is the test cross ratio obtained by the Mendel. So in his experiment of dihybrid cross, there is a difference between these two test crosses. So after writing these phenotypes, so we come to know what is the main reason for this deviated test cross ratios. This 7 becomes the parental type, that is blue and long pollen grain. Blue and long pollen grain. So this is the blue and round pollen grain. This is the red and long pollen grain. This one is the parental. This one is the parental, that is red and round. Red and round. Here the reason is the two dominant alleles or two recessive alleles present on the same chromosome do not segregate and assort into the different gametes. They tend to remain together and enter the same gamete. That is the reason. So why parent type percentage of parental type is more than the recombinant type. Here the more number of gametes produced. So in these crosses containing the dominant alleles and recessive alleles together. These two dominant alleles and two recessive alleles togetherly enters the same gamete. So this is the one cross. Then they did so another cross. So this is an example for copulation. The copulation means the dominant alleles or recessive alleles come from the same parent, enters the same gamete. Now go for understanding the repulsion by one more experiment did by them. Still the parent here taken were blue color round pollen, not long, versus red color long pollen. Their genotypes are like this. Blue means capital B, round means small l. Then red color means small b, long pollens represented by dominant alleles L. Then the gametes produced by this plant like capital B, small l. The gametes produced by this plant contain small b, one recessive allele, one dominant allele. So when cross is made between these two plants, the F1 generation was produced with the genotype of capital B, capital L, small l. So this is the heterozygous. This is also heterozygous blue color long pollen grains producing plant. Next, so again they went for test cross BBLL into take the homozygous recessive plant, that is small b, then LL, not this one, the recessive one, right? Recessive one. Then same the gametes produced by these are four types of gametes. By this one type of gametes are produced. Then see the test cross ratio. So in this cross it becomes 7:1:1:7. Just reverse 7 is to sorry. So 1 is to 7:1, right? So here the 1 is the parental type. This is a recombinant type, that is blue long, right? This one is blue round. This one is red long. This one is the red round. Right. So here also, so they obtained the different test cross ratio which is deviated from the Mendelian ratio. The reason is, so there is a less percentage of independent assortment of the genes or alleles. Here the genes are linked. Genes are linked and present on the same chromosome. So during the inheritance, the linked genes are so enters the same gamete. So because of that reason, they obtained the different test cross ratios. So in next video, we talk about the experiments did by T.H. Morgan and his colleagues. Thank you for watching this video.